Okay, we will come to our last recording. We'll be looking at the control of gene expression at the post-translational level. Alright, so post-translational level obviously means that translation has already occurred. We already have made the protein. Right, so some of us might think, right, we have already made the protein, right? Wouldn't gene expression has completed already, right? The gene has been expressed, the protein has been made. Why do we still have to look at what happened after translation? Right, so what happened is a lot of proteins, right, after translation, they may not be functional straight away, right? So part of post-translational control would be to regulate the function of this protein. So if the protein is not yet functional, right, after translation, then you actually don't see any changes to the cell after this protein is made. There's no expression of the gene in terms of the phenotype, right? So we have to make sure that the protein becomes functional for there to be some changes, some functions, right, in the cell for us to see the expression in terms of the phenotype, right? So that's why gene expression does not stop after the protein is already made. It stops, or rather we go on to look at how to control the function of this protein, make sure that it's functional when it's needed, then to stop its function when it's not needed, and also to break down and degrade the protein once it is really not needed anymore. All right, so this is what we'll be looking at the post-translational level. All right, so in terms of the control of protein activity or function after translation, we'll be looking at the biochemical modifications of proteins as well as proteolytic cleavage, and to control the amount of protein, to remove the proteins that are faulty or no longer needed, right? We will be looking at the process of protein degradation. Right, so this will be the three things we'll be looking at for this part of the lecture. Okay, so firstly for the biochemical modifications, right, we'll be looking at the addition of certain chemical groups to the proteins. Right, so I have a few examples in the notes. Okay. There are lots more examples. There are lots more different kinds of chemical groups that can be added to proteins. All right. So what I have included in the notes are some of those that you have learned or will learn examples of in the syllabus. So there can be a bit more linkage between different topics. No point I give you some examples that you will never see again, right? Okay, so when we add certain chemical groups to proteins, right, what's going to happen is it will affect the folding and final 3D conformation of the protein. Right, so you know that the 3D conformation of protein is tightly linked to its function, right? So when there is a change to the 3D conformation of the protein because of the addition of the chemical groups, then obviously it affects the function. Right, so a protein that was inactive, right, can be activated after addition of such chemical groups, or a protein that was active, right, can be inactivated by the addition of such functional groups. And of course, and once we remove the functional group, right, then the effect is reversed. All right, so this is how protein activity or protein function is regulated after the protein has been made in translation. Okay, so I'll leave you to read the examples in the notes, right? So the concept is really addition of chemical groups to slightly change or alter the conformation of the protein, and therefore altering the function and regulating the function of the protein or the activity of the protein. All right. A second mechanism of control of protein activity, this is really more for activation, right, would be proteolytic cleavage, right? So proteolytic just means proteins, like of proteins, and cleavage is to cut. Okay, so what happened is a lot of proteins, right, not the entire polypeptide sequence would be found in the final functional protein form. Right, there will be some trimming or some cutting of the polypeptide in order for the correct folding to the correct conformation, which would be active. Okay, so one very well-studied example is insulin, which you'll learn next year, if you make it to next year. So the insulin polypeptide rather, that is synthesized, right? What happens is there will be some cutting of the polypeptide, right? The signal peptide would be removed, and also the C chain, which is like somewhere in the middle of the polypeptide, would also be removed. The final insulin molecule, right, would only contain the B chain and the A chain, which are joined together via disulfide bonds, right? So if we don't cut away the unnecessary parts of the polypeptide, then obviously that affects the folding, right, of the polypeptide. So we will not get the correct conformation or active conformation of the insulin molecule. Okay, another example that you have learned before would be your pepsinogen, actually, in digestion, right? So in order for pepsinogen to be converted to the active pepsin form, right, there's also some cleavage of the polypeptide in order to expose the active site. Okay, so a bit more on the signal peptide. Actually, you have learned this before under protein synthesis. Right, so this is the same picture you have in the notes for protein synthesis. I have this in the lecture as well. What happened is for almost all, I would say, for all of the proteins that are destined to enter the rough ER, and then of course go on to the Golgi, and then after that either get inserted into membrane or are exocytosed or excreted from the cell. What happened is all of them, right, would have a signal peptide at the start of the polypeptide sequence. And so in the long polypeptide sequence that is synthesized via translation by the ribosome, right, the first portion of the polypeptide would be called a signal peptide that targets this polypeptide to enter the rough ER, right? So we have actually covered this before, so this is just a revision. Okay, so once the polypeptide has entered the rough ER, as you can see here, the signal peptide is removed, right? So this is basically proteolytic cleavage, right? The translation has already occurred, right, to form the polypeptide with the signal peptide, and then the signal peptide gets cut. So that's proteolytic cleavage, right? So most, if not all, of the proteins destined to enter the rough ER will have gone through this proteolytic cleavage, and of course insulin would be one of the examples of proteins that is destined to enter the rough ER, since insulin is secreted, right, from the beta cells of the pancreas, right? So that would go through the rough ER Golgi pathway. Yeah. So for such proteins, membrane proteins as well, right, there would be this signal peptide that would be cut inside the rough ER. So that's proteolytic cleavage. Okay, right, so last mechanism for translational control would be the control of the amount of proteins, right? So this is protein degradation of proteins that are no longer needed or that have become faulty, right? So there are different ways how proteins can be degraded. One of the more common way is what we call the ubiquitin proteasome system, right, whereby there is the addition of ubiquitin to the N-terminus of proteins that are to be degraded. Ubiquitin is a small protein, right? So this small protein will be added to the proteins that needs to be degraded via some enzymes, and this process of addition of ubiquitin is called ubiquitination. So a protein that has been added or tagged with ubiquitin molecules would be targeted to enter the proteasome. The proteasome is a very big protein complex which contains proteases, right? So it's not just a protease enzyme, it is a very big protein complex with a compartment with a space in the interior. So it's like a cylinder. It really looks like a cylinder under the microscope, yeah? So the protein that is meant to be degraded, what am I talking about? Sorry. So the proteins that are meant to be degraded would enter the space formed by this cylindrical protein complex, right? And inside there will be a lot of proteases, enzymes that break down proteins, right? So this tagged protein tagged by ubiquitin, right, would enter and therefore get hydrolyzed. Right, so this is a very common mechanism to degrade proteins in the cytoplasm, right, with the ubiquitination and the action of the proteasome. Okay, so I have a video that I've included in the SRS lesson, right, on this whole process. So hopefully this that video will give you a better understanding of the process. Okay, right, so that comes to the end of our lecture for this topic.